All right, let's start chapter 2, which is about microscopy. So, in this chapter, we're going to talk about the terms associated with microscopy, different types of microscopes, and how we prepare specimens for microscopy. The units of measurement that we use are kind of from large to smallest here, millimeters. That is something you could probably see pretty easily with your naked eye. It's about a period in 12-point font on a typed piece of paper. That would be too large for a microscope that we would use in today's labs just because they're highly magnified. Microns are things that are very comfortably within our range for using a microscope. The microns are things that are like cells or bacteria. Those are in that size range. It's a millionth of a meter, if that helps. But we also have some kind of new crazy microscopes that allow us to see things that are smaller. A nanometer is a billionth of a meter, and things that are nanometer in size are like molecules, viruses, proteins, and we actually have some even fancier microscopes that I'll talk about in this lecture that allow us to see atoms. In terms of the history, you should know Robert Hooke was kind of an important figure. He termed the word cell. Looking at plant cells basically with a magnifying glass, he kind of noticed that they have kind of rectangular shapes and termed this word cell. And it really is sort of the central understanding of biology today, like the cell theory, like in order to be alive, it has to be a cell and it has to have all of these characteristics and that kind of stuff. But if you think about it, we needed a unit of something to have in common, so we were all looking at the same thing. And this picture is so amazing. It is a picture of the first use of a microscope in medicine. And what I think is so hilarious about it is that the lady is holding the light and like some random dude off to the side is like pointing his microscope up at the sky, which is just kind of hilarious. But anyway, if you think about it, microscopes haven't been used in medicine all that long. First microscope was made by a Dutch eyeglass maker called Leeuwenhoek. And the principle of his microscope is really pretty the same as a modern microscope. It's just a curved piece of clear glass that's like a magnifying lens. And then he had a screw that was adjustable so he could basically control the focus. This picture from your book is great because you can kind of see the size range of things and what type of microscope you'd need to see them. You can kind of see millimeters, a frog egg. I remember seeing a lot more frog eggs when I was a kid, but they're approximately a millimeter. As you get a little bit smaller in the micron area, that is a pretty large cell. A red blood cell, for example, is 8 microns in diameter. Usually bacteria and the things we play with in this class are a little bit smaller than that, but they're still within the realm of a light microscope, like a standard lab microscope. But the smaller we get into the nanometer range, the more fancy the microscope needs to be. That would be like an electron microscope or some of the other fancier ones that we'll talk about later in this chapter. So back to the basic idea of a microscope. It's a lens that's curved glass that allows you to see something bigger and the ability to focus. Leeuwenhoek's microscope is basically in our compound microscopes. It's just we're fancier today. We have one magnifying lens in the eyepiece, the ocular, and we have a set of objective lenses that we can change the magnification of. And usually those are going to be 4x, 10x, 40, and 100x lenses. And what that means is that it is 4, 10, 40, or 100 times more magnified than you could see with your naked eye. So because there's 2 lenses, you have to multiply those together. And it's always good to note what your eyepiece lens is. The oculars are usually 10x, but sometimes they're 20. So a fancier microscope, you might actually get another little bump of magnification. But in order to calculate the total magnification that is happening in a microscope, you just multiply those numbers together. So if the ocular is 10, and you're using like the 4x lens, it would be 40 times magnification. Some important terms that I'll want you to practice with are relevant to microscopy. The first one is going to be resolution. Resolution is how clear an image looks under the microscope, and the better the microscope, the better the resolution. So you can kind of see at the bottom 3 blobs. The one on the left is a single blob, and then it's kind of conjoined in the middle, and it's 2 separate points on the right. A cheaper microscope is going to blur things that are nearby into one blob. That's pretty poor resolution. The better your microscope, the more physically separated different things are going to look, even if they're close together. So resolution is basically the power of the microscope to distinguish 2 nearby points. There's a few things that we can use to kind of enhance resolution. We use immersion oil. We can put a filter on the microscope that filters out certain colors of light that kind of brighten it up for our eyes, or we could just abandon light and use electrons. One of the interesting things about microscopy is it takes advantage of something called refraction. So refraction is what happens when light is passing through different types of dense materials. So for example, air is not dense, but glass is very dense. So as it goes from air into glass, it actually refracts, or it bends, and as it bends, we can actually focus that light into the center of our specimen on a slide, and then hopefully get that light up through the objective lens, through the microscope, and into our eye. That's what's going to give us clear resolution. One of the cool things about how immersion oil works is that it has, I know it's hard to believe, but it has the same density as glass. So in a preparation using oil, there's oil between the glass of the lens and the glass of the slide, and there's no like extra light refracting away from that image and making it appear blurry. So it's kind of this technique that is just really common in microbiology, and I just kind of want you to know that that's how immersion oil works. Like, it basically has the properties of glass in terms of refraction, and then it reduces the amount of light that basically bounces away and out of the microscope. Here's how a blue filter will work. Like, you'll kind of notice that your eyes pick up more sharpness when it's on the right. The blue tones are just easier to see the edge. This is the thing about the human eye. We just see certain colors sharper. So there's just a filter inside your microscope that's kind of artificially like enhancing the colors that you see so that you get a stronger edge and a stronger line in your eye. Okay, so for some different types of microscopes, I don't want you to go too much into the physics here, but I do want you to kind of recognize images that are coming from different types of microscopes. So the first is bright field, and it implies that the field of view is going to be super bright, like white light. So basically the light is going to come up from the bottom of the microscope, hit the specimen, and the specimen will be dark because it's absorbing or blocking that light against a white background. This is like your typical cheapo lab microscope style, is a bright field compound microscope. If you look at a dark field microscope, it's the exact opposite. Only the light that hits the specimen is going to enter your eye. So it's like a bright glowing organism against like a perfectly black backdrop. Phase contrast and differential interference contrast is kind of a special way of enhancing an image. It's a prism, actually. It's like a very fancy piece of carved glass that's inside the microscope. You wouldn't even know that it's there, but it basically enhances light that is coming through dense but clear structures. So for example, on the pictures at the bottom, there's the bright field, which is the pink paramecium against a white background. In the middle is the dark field, which you can kind of see, looks a little different. The membrane is kind of glowing. That's kind of cool. But the phase contrast on the right, you can really see the cilia around the outer edge. Those structures don't really appear anywhere else very well because they're clear and they're not colored. Phase contrast artificially accentuates the diffraction or the refraction of the light that's passing through those structures. So it can kind of like really pop out certain structures like a flagella or cilia or things that you might not ordinarily see. Fluorescence microscopy uses a special dye to basically glow in the dark against a dark field. So these are really cool. They're really bright. It is dark field, but because it glows, like, you see things really, really super easily. You should know that a lot of fluorescence is very specific to certain types of cell structures. And this is really helpful in medicine because we can fluoresce, like in this example, we can fluoresce the nucleus, or we can fluoresce the microtubules, or we could fluoresce something like breast cancer cells. Like, fluorescence microscopy, it makes diagnosis of a microscopic specimen like super easy because it'll just glow in the dark and it only attaches to certain specific molecules. Here's some more examples of fluorescence. It's just really pretty. I could look at these pretty much all day. Confocal is a type of fluorescence basically that uses a laser or light to basically get deeper, like, structures illuminated. This gives you a sense of depth. Like in this particular picture, you can see these two kind of star-shaped structures inside the paramecium. Those are actually contraction vacuoles that allow it to squirt water out if it wants to rise or sink like a submarine. You don't see those in any other type of microscopy. And what I think is really good to understand about confocal is that it gives you depth, or a three-dimensional image where everything else kind of just looks like it's the surface of the structure. When you're getting into the higher resolution stuff, this is really exciting microscopy. But I want you to know that there are types of diseases that are actually diagnosed with a specific picture of it. Like, you can't just look at symptoms or whatever. Like, we'll have to actually take an electron micrograph or a picture of it. And these are really exciting. These are fancy. They've been around for a while, but, you know, I think that this is in some ways some of the more exciting things about biology, is being able to see this stuff. We'll start with electron microscopy. There's two types. The first is transmission electron microscopy, and this is where electrons are shot at a specimen that has been sliced. That's very important. It has to be sliced really thin, and it has to be stained with super heavy metals. So those heavy metals could be like gold, lead, cadmium, all sorts of weird stuff, but the metals do distort it a certain amount, and because it's sliced, you should expect it to look like you're seeing the interior of the cell. So this is the paramecium again. It's super amazing resolution, really clear. And you can see all these things inside the paramecium that we haven't been able to see before. There's like lots of little blobs. That is a classic way of viewing a transmission electron micrograph or a picture from a transmission electron microscope. Usually we just abbreviate that TEM, but absolutely beautiful. The apparatus is pretty big. We don't have one at Mount Hood. PSU has a really fantastic electron microscope lab if you ever are interested in doing more microscopy. But one of the things I do want you to know is that all electron micrographs are actually black and white. So here's kind of a variety. They don't look as exciting. When you see a colored version, it has been photoshopped, and they really don't actually have any color. It's just artistic. And this is a beautiful little bacillus with blue flagella swimming around. Compared to the Vibrio with a single flagella, it doesn't look as exciting, but that's actually the real deal. All of these guys are going to be black and white, and then an artist has basically created that beautiful color. Scanning electron microscopy is the second type of electron microscopy, and what this does is it just scans the surface. So look at this paramecium. Like, you can see all of these little curly cilia on the outside. We haven't seen its little hairs at all. So this is like a surface scan, and it gives you the absolute perfect vision of the outer three-dimensional surface. Here are some SEMs, some scanning electron micrographs. The one on the left is actually Staphylococci. It's got like purple glue that basically holds those little cocci into a staphylo arrangement. Looks really sticky and pretty gross. There's a beautiful spirochete on the top right. That spirochete actually comes from gingivitis, little inflammation of the gums. And on the bottom you can see the surface of a grass pollen, which just looks evil in my opinion. But like you can really see the surface structure. It's so cool. All right, in newer microscopy, we have some that are even better than electrons in terms of resolution. This would include the scanning, tunneling, or probe microscopy. I've heard it referred to as tunneling or scanning probe. Either way, same idea. How it works is it drags a very sharp metal tip over the surface of a specimen, kind of like a record player. And as that tip is moving up or down, a computer is going to generate an image based on how it's moving. And this is giving us incredible small detail, like 1/100th of an atom. But this is not actually approved for clinical use yet. It is still sort of research stuff, but it's cool. So here's just a close-up of the tip. It's going to drag over the surface of the specimen. And you'll see two generations of a scanning probe image. These are both masses of flagella, basically. And you can see it kind of looks digitized, like it looks like a computer did it. But this is incredible in the detail and the smallness of things that we can actually see. For example, if you just relax your eyes for a second, this is DNA. This is the first photograph of DNA. Like, all you have ever seen before is some artist drawing what they think it's supposed to look like. And for a lot of biologists, like, this is really revolutionary because we can actually see it. And these are just some cool things. This is on the left is pectin from apples. A pectin is like if you make jelly or jam, you've used pectin as a thickener. But what's really cool about pectin is that it surrounds microbes, and it actually makes it easier for your immune system to dispose of pathogenic bacteria. So it is actually true that an apple a day keeps the doctor away. And the one on the right is just little rings of genetic material. It's just so cool to actually see that stuff. Atomic force is very similar. It's just a diamond tip, and diamonds are hard. So the big problem with this type of microscopy is you don't know how your tip is staying sharp. If it's gotten dull like a pencil, you're not going to get the same result, which is why this is still kind of very theoretical type of microscopy. But this basically uses a diamond instead of metal, and it gives you just these beautiful three-dimensional images. A little more pricey though. Classy fish is really interesting, and I want you to take note of classy fish. Classy fish is what happens when you want to take an image of actual nature. So usually in science, we take an example out of nature and we simplify it, we purify it, we make it just one thing, and we take a look at just one thing at a time. But what if our question is like, well, we want to know like how diverse your oral bacteria are in the mouth? Because one of the interesting things about mouth bacteria is only about half of them will grow in a lab. The rest of them need to be in a human mouth. So if you really want to know what's actually there, you need to take a picture of them in the mouth. And that is what classy fish is for. It is dark field. It is fluorescence. But what it does is it allows you to take a snapshot of nature so that you can see the true diversity without trying to isolate and figure out what everything is and oversimplify it, because a lot of bacteria don't like to grow in our standard lab conditions. And this is actually a classy fish of the oral microbiome. There are at least 700 different species in the mouth. That includes some archaea, and only about half of those bacteria even have a name. And it's because we just can't work with them yet. So classy fish kind of is that other missing question for us, is like, how do we know what's actually in the real world? And then I watched this really great TED Talk, and since you guys are trapped at home and there's nothing better to do, you should watch this TED Talk. It's so cool. It's this origami microscope that's made out of a piece of paper, and it only costs like $2. I thought about having you get some for Amazon just to do for lab, but I don't know how safe Amazon is and all that kind of stuff, so I didn't actually go for it, but it's really cool. It's just a piece of paper that you can like take on the field or send it in a suitcase. Like, one of the things about field microbiology is these microscopes are really delicate and you can't just like drag them out into the wilderness. This is going to change everything about medicine. It's just going to make microscopy really cheap, and it's indestructible. And it's cool. It's like literally a piece of paper with a lens, and it only costs like $2. Okay, now just some things about specimen preparation for microscopy. This will have some lab activities associated with it too, so you'll be able to watch this preparation and think about it. But first thing you do is just get your bacteria on your slide, and that does require that you transfer them, and then you pass the slide through flame, which causes the bacteria to stick to the slide. You just don't want to overdo it because you're literally not cooking it, you're just kind of sticking it to the slide. And then once you have it stuck to your slide, you can add all sorts of chemicals which are going to produce colorful contrast. So contrast is another important term in microscopy. It's for your eye, it's lightness and darkness. So contrast is like what we need to add to a specimen that doesn't have any natural color so that we can see it against a white background. And we use a lot of different colors. They all have different chemical properties, but it's important to know that it isn't really that color. We've been adding all of these just historically so that we can see something under the microscope. Couple different types of stains that we're going to learn about in this class. Simple stains, you just put some color on it and then it's just, you can see it. It doesn't really tell you much, but at least you can see it. Those are not super useful in clinical stuff, but it's a great place to start just to kind of learn how bacteria look in terms of their morphology or their shape or their arrangement. The differential stains are more important, and these are going to tell you something about the bacteria based on the results, and some of those stains will be like the gram stain, the acid fast stain. We'll talk about those in a second. And then we'll have some cool special stains that you'll learn about that will be developed for looking at a particular structure or feature of bacteria. For example, if it makes an endospore, like that survival cyst, that is something that is very relevant. So we have a special kind of technique to know whether or not it has that property. Simple stains use basically a chemical attraction for how they work. What you'll see is that a bacteria cell has a negative charge, and most of these chemicals have a positive charge, and those two things attract. So the positive color gets inside the bacteria, and then that's what you see. And it's all one color. It's either going to be red, purple, blue, whatever. Those are how simple stains work. If you hear the term mordant, mordant is a special chemical that we add to certain types of stains that like enhance something, or it makes some chemical property change and makes it work. Mordants are often really large molecules, like iodine, for example, is a really big molecule. It's element 52 on the periodic table, and whenever you add anything that big, it's going to coat things and make them look a lot bigger, which can be helpful for us. But think of a mordant as like a special ingredient that enhances a stain. This is a good table from your book that kind of gives you some examples and like what the different chemicals are, but basically just think of a chemical attraction for simple stains. If you hear the word negative stain, negative stains are like in art where you color the negative space. Basically, it'll be a chemical that does not get into the bacteria, but it stains all the background space, and it's like the opposite. Like the bacteria will be clear or transparent against a darker background, but those can be really cool to look at too. Differential stains, this is kind of where the money's at in micro. Like, these differential stains will basically tell you something about the bacteria. And I'm going to start with an explanation of the gram stain. So the gram stain is going to basically identify two types of bacteria, gram-positive and gram-negative. Gram-positive bacteria are the purple ones, and in this case, they are streptococci. And the gram-negative bacteria will look red. And in this case, they look like bacilli. So a little bit more of a rod-shaped. So, like, it's literally purple or red are the results. And it's, if it's done well, really obvious to tell the difference between gram-positive and gram-negative bacteria based on color. But the color is actually the result of how these bacterial cell walls interact with those chemicals in the stain. So gram-positive bacteria have a really thick cell wall, and gram-negative bacteria have a really thin cell wall. And what's weird about gram-negative bacteria is they have an outside membrane in addition to their normal membrane. So they're very anatomically distinctive, and the gram stain is the most important thing we do in bacteria identification because it tells you so much information about the anatomy of your bacteria. We'll talk a lot more about the gram stain in the next chapter, so we're not done. Acid-fast staining is really for clinical significance, and that's important. Acid-fast refers to a certain type of bacteria that are in the family Mycobacteria, and that includes tuberculosis. So there's a few other things that are in that family in addition to that, but tuberculosis is so important. We actually think there's about 2 billion people on this planet that are infected with tuberculosis bacteria. It's in a latent form, but it's one of the really significant diseases on planet Earth for people. So acid-fast staining basically helps us identify tuberculosis. And the way that that works is that tuberculosis has a real waxy outer structure like around it. Like, it's really got the sticky wax. This chemical, it penetrates that wax and it stays in there even after you decolorize it with acid, which for most bacteria, it would just completely rip them apart. So tuberculosis is really interesting because it has like a very chemical resistant like structure on its outer walls that with the stain, it just looks bright pink. Those pink cells in there are the tuberculosis bacteria or the acid-fast positive bacterial cells. Endospore staining can come in a lot of different ways, but I think heat is probably the fastest way to get bacteria to like stress out, basically. This is really typical for soil bacteria to endosporulate when the soil dries or maybe it doesn't have the right environment. But they basically have the ability to just compartmentalize all of their important little parts of their cell into this little survival pod. And in this specimen, you can see that there's some red streptobacilli. Those are bacteria. But the little green circles are little endospores that formed from bacteria that were so stressed out that basically they formed an endospore and then they disintegrated. So this is another kind of Christmas colored slide where you can kind of really see the difference between the green and the red, and if you see the little green circles, you know that this is capable of endosporulating. Endospores are a clinically significant finding for us because endospores can survive usual disinfection protocol, and some of the nastier microbial infections out there, like C. diff, if you've ever heard of C. difficile, it can cause really bad diarrhea and colitis, basically. That endosporulates. And what's really interesting about C. diff right now is we've now documented that it can survive, the endospore can survive hospital laundry, which is frightening if you think about it. Like, endospores are just important that we know that they have this survival ability. Capsule staining is kind of a cool, like, extra special stain. It's a negative stain. It's going to stain the negative space. But what it really does is it illuminates the outer structure of bacteria. And we're going to talk about this more, but there's kind of these two options for bacteria to have like a sticky layer around their outside. It can be tight like a capsule, or it can be really diffuse and loose like a slime. So like your dental plaque, for example, is a good way of thinking of a slime. Kind of just like sticks to the surface of your teeth, and it's very disorganized. But those types of structures are highly relevant to us clinically because they allow for the attachment to mucous membranes. So that means that they can be potentially pathogenic, and the presence of a capsule is very illuminated in this type of stain as like a clear halo if it's a capsule, or it would look like slimy, kind of loose, gross slime if it was a slime layer. But that's the last sort of special stain that we'll talk about in this chapter.